A very warm welcome to all of you joining us today for what I know is going to be a fantastic, engaging session. In drug discovery, the decisions made earliest in the process can have an outsized impact on everything that follows, and yet they're often made under significant time and resource pressure. Today's talk is designed to help address that. Whether you're new to early stage discovery, evaluating how to get more from your screening strategy, or looking to de-risk your program from the outset, today's content will offer a practical, expert-led guidance that you can directly apply. We'll be drawing on deep expertise in medicinal chemistry and library design to explore how to identify and progress high-quality hit-finding starting points, from screening library selection right through to early hit evaluation, and how stronger decisions at these stages can meaningfully reduce downstream risk. An expert who understands this space and how to get the most out of it is Sam. Sam Ceusters is Head of Chemistry at Symeres proprietary Cymagold compound screening library. His role focuses on the design and development of novel chemical libraries to support small molecule drug discovery programs. He has a background in medicinal chemistry and drug discovery research, and Sam works closely with biotech and pharmaceutical teams to help identify high-quality starting points for drug discovery campaigns and accelerate hit identification. So without further delay, I'm delighted to hand over to Sam.
Well, many thanks for the introduction, Katie. Today I will be talking indeed about the greatest hits, a beginner's guide to high-quality starting points for drug discovery. I've condensed a lot of information in about 40 to 50 minutes, so buckle up, enjoy the ride. If you have any questions, as Katie noted, feel free to ask them during the Q&A session. I have numbered my slides in the bottom left corner of my presentation, so feel free to refer to a slide number if you have any questions. And then let's crack on. So I'm pretty sure you've all seen this graph in many shapes and forms and colors before. We're looking at the drug discovery to commercialization pipeline. And as Katie really nicely described it, there's a lot of decisions that you need to make, right? And we're quite often deciding whether to advance a compound or not. And the numbers change as you progress along the pipeline, right? Quite often we'll start by screening vast amounts of compounds, sometimes up to the millions. And as you progress to the hit to lead optimization stage, you'll be making hundreds of analogs to end up with 2, 10 clinical candidates, which eventually, hopefully, turn out to yield one FDA-approved commercialized drug. See, every time when you apply a selection criteria, that's a decision that you make. And of course, you want to make sure that you make the right decision at the right time, because it wouldn't be the first time that you fail in the clinical candidate stage or fail after a few years. But we all know that this entire process takes years, if not decades, and the longer it takes, the more resources it costs. So making the right decision is vital, and it will save you a lot of time and a lot of money. So that's definitely what I want to help you out with today. Today, I will be focusing on the decisions that we make early stage drug discovery, more specifically around hit generation. Hit generation is focused on hit identification first, followed by hit validation. And with hit identification, I'm pretty sure once you perform your primary assay in search of hits, that you've thought about the biological relevance of your readout. But I also want you to think about the interference that you may observe in the readouts of your assay. You see, if you perform an assay and you're based on a fluorescence readout or a UV absorption readout, be aware that the small molecules which you're probing, that they may also fluoresce or may also absorb at those wavelengths. So that's something to keep in mind, because that can cause false positives or false negatives. Once you've then identified your hits, active compounds in your primary assay, you'll want to validate those hits. You'll want to perform counter screens, orthogonal and secondary assays. And what's also very important is you want to make sure that the structure, the purity and the stability is confirmed. You see, it wouldn't be the first time that it turns out that there's a small impurity present in your screened compound, and that impurity is actually responsible for the activity which you're observing. So keep that in mind. Now, this presentation wouldn't be called the greatest hits if I didn't include at least one musical reference. There's quite a lot of parallels that we can make, and maybe the most striking one, of course, is that both musicians and drug discoverers are looking for hits. So what makes a hit then? Well, just a hit. That's something like Never Leave You by Lumidee. And I bet most of the people in the audience have no clue who Lumidee is. But once I start singing that song, and don't worry, I'll save you for my singing skills, but I'm pretty sure you'll remember the tune. You see, Lumidee had quite a good hit in 2003, was played on radio for a year, but all the other songs which he released were not as successful, and so Lumidee was left to be forgotten after 2003. Now, a validated hit, that's something like Belgian techno anthem Pump Up the Jam by Technotronic, 1989. Certified banger, still manages to fill dance floors across the globe. No doubt, absolute amazing song. But we also need to acknowledge that all the other songs which were released by Technotronic weren't as successful. They weren't proper hits. So that's where Pump Up the Jam was left. What's then a high quality hit? Well, that's something like Bohemian Rhapsody of Queen. You know, meticulously crafted, highly curated, massive musical value. And humanity continues to reap the benefits of this masterpiece, right? Most importantly, all the other songs that were released by Queen, all other analogous songs, were hits in their own right. They're all timeless masterpieces. So what I want to help you out with today is how to identify the Bohemian Rhapsody of your compound screening collection. The musicians in the audience will probably be well aware of the 10,000 hour rule. You see, the 10,000 hour rule states that if you want to become an absolute expert in your field, that you need to have spent at least 10,000 hours perfecting your skill. And that's exactly what Ed Sheeran did. When he started off, he vowed to write at least one song for every single day. And he did that consistently for years. And after a few years, he found out what works and what doesn't work. And clearly, he's now very successful. But step aside, Ed Sheeran, because before you could even spell your name, our director of analytical and medicinal chemistry, Rutger Vollmer, was already in the lab, cracking on with the hard organic chemistry challenges and solving any medicinal chemistry for his company and the client. So Rutger has easily done his 10,000 hours, but of course, if you combine his experience with over 250 organic chemists, parallel chemists, medicinal chemists at Symeres, you could argue that we've jointly done well over 10,000 hours, and that you can easily trust us when we tell you that we know what works and what doesn't work. Now back to the molecules. What makes a hit? A hit is just an active molecule in your primary assay. A validated hit, that then has the confirmed chemical structure, stability, and purity, which we've talked about earlier, but it also shows a dose-response activity in primary and secondary assays. Furthermore, you've confirmed target engagement, which means that you're sure that that molecule is engaging with that target and not an off-target, say. A high-quality hit then shows all the properties above, but it also has attractive properties for further optimization, more specifically early ADME, the absorption, distribution, metabolism, and excretion of a compound. It has a relevant affinity for your target of interest. In other words, the concentrations at which it's active is relevant for the dosage that you intend to give. The compounds are chemically stable, which helps in the synthesis, but also the storage of these compounds. Ideally, a high-quality hit is novel. I'm not saying that they can't be novel, but I do urge you to properly assess any challenges that you may face when you're diving into very busy, very highly populated IP space. Right? And what's most important for me is that a high quality hit is not a singleton that you observe active analogs. You want to have initial insight in the structure-activity relationship, or shortly SAR. You see, if you have active analogs of a compound available in your set, it indicates that the activity which you're observing is embedded in the central core, in the central scaffold, rather than all the decorations that you put around. Now, okay, you've done your screening, you've identified a hit. Will that hit, that exact same molecule, will that molecule end up being the clinical candidate? In most cases, absolutely not. You see, a hit molecule is very likely to undergo multiple structural changes to further optimize its properties, both potency and ADME-wise. So that indicates, and that means that you will have to synthesize a lot of analogs. And we'll do that in the hit to lead stage and the lead optimization stage. And to give a bit of an idea of how that goes, I came up with this entirely fictitious, oversimplified graph. But the story goes as follows. You see, we've done our initial screen. We've identified 3 hit molecules, and all these 3 hits are structurally distinct. They come from a different structural cluster. One thing to keep in mind is that hit molecule number 1 is easy to synthesize, so we can quickly follow up with structural analogs. Hit molecule number 3 is hard to synthesize, so it'll take you a lot of time before you can make your target molecules. So we dive into the lab, and first thing we do is we make small structural changes. We do that for 2 iterations, and it turns out that some of these analogs are active, some of these lose their potency. And at this point, the molecules that lose their potency are deprioritized because, at all times you're looking at limited time and resources, right? Now, after 2 iterations, we decided to look at all the insights, all the data that we got, and apply those insights to future structural changes of this inactive analog of hit molecule number 1. And look at that. In the 3rd iteration, that yielded 2 active analogs. And in the 4th iteration even, we managed to make a lot of analogs of hit molecule number 1 because we could do so, because it was easy to make. And also, you can see here that we stopped making analogs of hit molecule number 3 because the 3rd iteration gave, both inactive compounds, and we deprioritized it at that point. But once again, after 4 iterations, we got a lot of extra data, we got a lot of extra insights into what was allowed and what wasn't. So we once again decided to apply those insights to the latest active molecule of hit molecule number 3, because series number 3 actually had really nice properties in terms of ADME and physicochemical properties, but it just wasn't active. And when we applied those findings, we generated 4 active analogs. And so this is what we ended with. We ended with 9 compounds which were favored, which we found really interesting, and these were advanced to further in vivo testing. Furthermore, we also had multiple backup compounds. You see the color is slightly vague, but all these other active analogs, we could also progress with those. We didn't, because you have to make priorities, but we have a plan B, a plan C, a plan D ready in case these 9 compounds fail. Now, what are the lessons that we can learn from this? Well, first of all, if you want to gain insight in the structure-activity relationship data, you need to make analogs. You need to test structural analogs. Now, the insights that you get from that initial SAR data, that can be transferred between compound classes, because as long as the compounds engage with the same catalytic domain, the same binding pocket, you can transfer those insights. That's what we did when we made a bunch of analogs of hit molecule number 1 and applied those findings to hit molecule number 3. And that brings us to the 3rd point, because multiple clusters create those options. They allow you to transfer those insights, plus you have your backup possibilities. You see, when a lot of money is involved in your decisions, you want to have options. You want to bet on multiple horses. You can imagine that if we had put all our money only on hit series number 2, that we could have stranded here. And that's why hit molecule number 1 was so interesting, because, once again, we could transfer those insights. I also want you to keep in mind, and this is very important, that every single point in this graph represents a physically synthesized molecule. Someone went into the lab, spent time in the lab to make this compound. So synthetic feasibility is key. If you can't make it, you can't test it. Now, before we dive into the different types of screening libraries, I want you to think about what you have in mind already. What are you looking for in your drug? What are your desired properties? Because you can, look at all different types of screening libraries, but you need to have some criteria that you want to hold against them. And these criteria can then be a very useful guide, right? See, you can look, for example, at desired drug properties. Do you have a preferred administration route in mind? Are you targeting an oral drug or an IV drug? Do you want your your drug to penetrate the blood-brain barrier? Or do you exactly not want that? Are you looking for covalent binders or PROTACs? These can all help you guide your search. In the same way, we can make the distinction between a known target and an unknown target. See, with a known target, there's quite often a lot of information available already, right? For example, X-ray structures. And an X-ray structure gives us very rich information about the key interactions that your molecules need to make, and also the shape and the nature of the binding pocket. These molecules were, by the way, synthesized by my colleagues. So you can use that information to already get to a preferred pharmacophore or identify favorite motifs. So you can do substructure searches once you have your hands on a commercial catalog. If you don't have this information available, you could maybe want to look for substrate analogs. You can imagine that if you're the first person in the world to target a kinase, for example, that the first thing you may want to do is to look for ATP structural analogs, right? Now, with an unknown target, quite often you'll be performing phenotypic screens because you're looking at a desired phenotype, but you don't yet have the target in mind, or you don't know yet what your target is. So then you can approach it in two different ways. If you're focusing on just identification of your target, a tool compound may be enough. So that could imply that all the interesting ADME governing physicochemical parameters may not be as important, and then your focus may be on maximizing the amount of initial hits and then just compare all those structures. But if you're interested from the start in a drug which you want to further develop and turn into a marketed drug, it may be interesting to already limit your selection to favorable physicochemical properties. So those are things you can keep in mind and use as a guide. Today, I will talk about 5 types of screening libraries, the major types. First one is a DEL library. We'll talk about fragment-based drug discovery, virtual libraries, targeted libraries, and diversity libraries. A DEL library is short for a DNA-encoded library, and it works simply as follows. We're screening a large set of small molecules, but all these small molecules are tethered to a DNA linker. And the sequence of this DNA strand encodes for the structural information of the small molecule which it is attached to. To put it simply, the top 2 molecules here have a blue triangle in the top left corner of their structure. Well, the terminus of this DNA strand encodes for this molecule has a blue triangle on the top left corner of its structure. Having this information attached to the small molecule enables you to screen all your small molecules, your entire set, at once. You can all add them in one vial to your desired target, and it allows you to perform an affinity-based assay where you wash away all non-binding molecules, you retain the binding ones, and then you can perform PCR to amplify the DNA strand that's linked to your binding molecules, and then sequence that DNA to extract your structural information. One of the major pros with the DEL library is that it allows you to screen a vast amount of compounds in one go, in one experiment. And the fact that you can generate big data in that way also makes it compatible with AI and machine learning. You see, machine learning can help you identify trends that you wouldn't have observed with the naked eye, or it can also help you deconvolute that interesting data point which would have otherwise been lost in the noise. Now, a trade-off for getting that big data is the fact that you're, of course, gluing essentially a small molecule to a macromolecule. So you will inevitably have steric hindrance of your DNA linker. To put it simply, the chance of this purple hexagon binding deep inside the binding pocket of a target of interest is very small because you have this entire DNA strand that's just blocking the entrance. It's just in the way. Another thing that you may want to keep in mind is that the output data of a DEL library is different from other libraries. Your output here will be enrichment. And enrichment is not always linearly correlated to KD or activity values. What you'll also have to do with the DEL library is, if you want to validate your hits, you will have to resynthesize them without the DNA strand. And then once again, when you do that assay, you still need to generate that KD or activity data. Fragment-based drug discovery goes to the other side of the spectrum. We're looking at very small molecules. We're looking at fragments, in fact, with a very low molecular weight of lower than 300 Daltons quite often. We'll start by identifying binding fragments via SPR, NMR, X-ray, mass spec, TSA. And once we've identified a few, we'll either grow, link multiple fragments, or merge multiple binding fragments. This way, we're very efficiently sampling our chemical space. We're generating hits with high ligand efficiency. Why? Because we are growing and making our molecules very intently. You know, when you're screening just a set, there's a chance that you'll have structural redundancy, so that there's a decoration on here that does not actively contribute to your desired properties or your binding. And the chances of having that redundancy in a fragment-based drug discovery approach is rather small. Now, a con of FBDD is the fact that when you're looking for binding fragments, that you'll initially be looking at very low KDs, well, you know, very high concentrations. So you'll be looking at the millimolar, micromolar range. So that can imply that your detection may be challenging and that you may also observe quite a lot of false negatives. The other thing is that D is a chemistry approach. If you're growing, linking, or merging your fragments, you will be performing additional chemistry. So that will once again take time, and therefore money. A virtual library speaks for itself. Virtual, you do it on a PC. You can generate a lot of data in a very short time frame, right? High throughput. But the synthetic tractability is not always guaranteed with these molecules. You know, in the lab, we tend to say you never really know if you can make a molecule unless you've actually made it, because otherwise we'd just be glorified bakers and not scientists. The other thing that I want you to be cautious about is that numbers can be deceiving. You see, say that we take 5 core structures, 5 scaffolds, and we decorate them with literally any molecule you can possibly think of. Well, then you can easily get to a set of 5 billion compounds, but is that really structurally diverse? You know, those 5 billion compounds will all depend on that structure of those 5 cores. So look at the structures, try to assess that diversity yourself and look at it from a structural point of view. The other thing is, of course, yes, you'll be generating a lot of data in silico, but you will need to experimentally validate. And that's, of course, when synthetic tractability becomes important. But keep in mind that you'll also generate a lot of false positives. But, you know, this is under major development and it's increasing yearly. We're aware of that as well. But this brings us to the more traditional, but still gold standard approaches, which are targeted libraries and diversity libraries. Both libraries have the physical molecules. You know, these are the hit molecules that you're testing. These are the molecules that get you that KD data, that very relevant activity data. And a targeted library is then focused on a predefined chemical space, right? So we're looking at a kinase library or a CNS library. So that chemical space is already quite narrow, and you'll quite often have a lot of information already at hand about the binding mode, SAR properties. With a lot of information often comes a lot of competitive IP. So that's one of the challenges that you'll likely face with a targeted library. So you may have to be quite creative to get out of that competitive IP space from a synthetic point of view. And also, especially when you're targeting enzymes with a highly conserved binding site, then selectivity may also be one of your major challenges. Diversity library is then focused on broad chemical space coverage. It's rather unbiased. So that also increases the chances of finding IP-free space, right? It leaves room to find novel molecule target interactions or mechanisms of action. Or identify novel targets, right? But if we're talking about novel target and identifying. So in a phenotypic assay, yeah, that can be difficult. And if you're performing a phenotypic assay or a wholesale assay, that's also more expensive than your classic in vitro experiment. Now, okay, you've had a look at the selection of compound libraries, and you've made a decision, and now you have a set of molecules in your hands or on the PC on paper. How do you now assess that compound collection, right? And more importantly, well, equally importantly, once you've run your assay, how do you look at your hits? How do you assess them? Well, I'm going to use a diversity library as the example because it's quite rich actually. Because with the diversity library, quite often we're looking not only for new hit molecules, but also for new targets. And if you don't know where you're looking for it, it feels a bit like shooting a bow and arrow at a target, but you're doing so blindfolded and you have absolutely no idea where your target is. So let's do so, start shooting. And you'll see that the recreational archer may shoot at random. And when they take their blindfold off, they'll notice that they've covered some corners of the room, but definitely not all corners, and they've missed the target. A skilled archer, on the other hand, may have a very high accuracy because they're very used to aiming at a certain corner of your wall, but they were aiming at the wrong corner. And they take the blindfold off and they notice that they've missed the target. All right, so what would be the ideal approach? What approach would maximize your chances of hitting that target? Well, if you have a predefined space, if you know how large your room is, you could argue that you may want to cover every corner of that room evenly in almost, equally distributed manner. And that would, of course, increase your chances. Now, when we're performing drug discovery, of course, we're not sampling 3D room space. We're not archers. We are looking at chemical space. And chemical space can be defined in terms of whichever properties you're interested in. If you're looking at a 2D graph of chemical space, you could say, look, we're interested in molecular weight and lipophilicity. These are the ranges that we would like to investigate. Well, then a perfect diversity library could look something like this, where every dot represents a molecule that has those specific combinations of log P and molecular weight. But once again, the space which we're probing in drug discovery is far larger than just 2D space, right? We're looking at multi-parameter optimizations, a multi-dimensional space, and it's also a subset of the entire chemical space. We are looking at the bioactive small molecule space quite often. And that means that we're looking for molecules that have a favorable ADME profile, which is governed by physical chemical parameters such as molecular weight, lipophilicity, polar surface area. And you also want to have both selective and potent molecules. So your selectivity is quite often governed by your hydrogen bond donors and acceptors. And the potency is influenced by hydrophobic groups. And then, of course, in order to make sure that those polar groups make those key interactions, shape and size of your molecule is essential to facilitate that orientation, right? So when we look at physicochemical properties, I'm pretty sure you've all heard of Lipinski's rule of 5. I'm not going to go too deep into that. Nowadays, there are plenty of other selection criteria as well, such as like the lead-like fragments criteria. And Weber's rule is often noted in the same sentence as Lipinski. There are 3 things which I want you to remember. First of all, these are rules of thumb. And Lipinski is a rule of thumb for orally bioavailable drugs. There are plenty of marketed drugs which exceed the rule of 5 or which violate more than one criterion of the rule of 5, and yet they're perfectly fine drugs. So don't take this as a hard limit. Keep it in mind. Take it as a soft guide, if you like. I also want you to keep in mind that all these rules pose upper limits. And that will be important for a slide later on. And how do I want you to apply these rules in practice when you're looking at hits? Well, if you have 2 hit molecules coming out of an assay and they have comparable properties, comparable potency, I want you to favor the low molecular weight low lipophilicity compound. And why you need to do that will be explained later in this presentation. When we talk about shape, quite often you'll see a PMI plot. And PMI is short for principal moment of inertia. And depending on the shape of your molecule, it will either end up in the linear, disc-like, or sphere-like corner of the chemical shape space. This particular graph is a random sample of the Campbell database of bioactive small molecules. And what makes this graph so interesting is the fact that we see a bias towards flat molecules. Why is this interesting? Well, when you look at the structure and shape of natural products, molecules that have been optimized for over millions of years, often with high potency, high selectivity, well, a very large portion of the natural products reside in this corner of chemical shape space. So that indicates that there's still plenty of opportunity to grasp by venturing more into the sphere-like chemical space. How can you do so? By introducing more sp3 carbons. What does FsP3 mean? It indicates the relative amount of sp3 carbons in a molecule. And for the people who haven't studied chemistry, this all has to do with the hybridization state of your molecular orbitals around your carbon. I'm now realizing that that doesn't say anything either, probably. In practice, an sp1 carbon engages in a triple bond, yielding linear molecules. An sp2 carbon engages in a double bond, yielding more plane-like structures, and an sp3 carbon adopts this tetrahedral orientation where every binding partner sits in one of these corners of that tetrahedron. And when you compare structures, you can see that the complexity, the diversity, and quite often also the novelty of your compound increases when you increase your amount of sp3. You can look at this molecule, for example, and if you swap this methylated nitrogen with the methylene bridge here, you turn it upside down, that will give you a completely different structure that will fit completely differently in a target. So that will have definitely an influence on the selectivity. And the same with that proton here. If we flip it up, there's quite a big chance that this entire ring structure will fold completely differently to reach that minimal energy. Now, in 2009, Frank Lovering and his co-workers, they published the seminal Escape from Flatland paper. And in that paper, they observed that as a molecule progresses through the drug pipeline and advances from hit to lead and clinical candidate, that the amount of sp3 increases. So Frank Lovering made the argument, why not start with sp3 rich molecules to begin with, right? He also noticed that as your FsP3 increases, that also your solubility and your selectivity increases. So there's a lot to gain. Furthermore, we also know that flat molecules often tend to be more promiscuous and thus tend to give more false positives. And to put it, practically, you can also imagine that one flat hit single has multiple binding pockets, and it will just as easily fit in this binding pocket. In other words, every toothpick will likely fit in a door, but you won't be so likely to open that door with a toothpick unless you are a professional lockpicker. You'll often hear us talk about SAR as well. And we'll say this molecule series has poor SAR, or it has limited SAR, steep SAR, or good SAR. And what do we mean with that? It all has to do with how the structural changes in our molecule influence those desired properties. And if you have a poor SAR, a slight change to your molecule will often result in a loss of your desired property. And if you look at that visually, you can imagine this blue shape to be the chemical space that you're covering with all the analogs that you've made of a single compound. And you then see that there's actually a small overlap between that covered chemical space and the desired property space. Good SAR then implies that that overlap is rather large, right? It doesn't necessarily need to mean that you have complete overlap with your desired property space, because it's good to know what works and what doesn't work, but it does give you options to further optimize your properties without losing everything with, you know, a small change, or at least not if you're here rather than there. So that's what we're looking for. That's what we're looking for when we talk about good SARs. It allows us to tweak the properties, really. How do the hit, lead, and candidate molecules look like in terms of chemical space? Well, you can see as your molecule advances through the pipeline, that molecular weight, lipophilicity, FsP3, amount of rotational bonds, TPSA, and hydrogen bond acceptors tend to increase. And remember when I said earlier that Lipinski's rule and all those criteria, that they pose upper limits? This is why that's important, because, you know, especially with molecular weight, you can imagine that if you start too high with molecular weight and you're looking for, you know, a small pocket, at one point, your molecule will just become too big and it won't fit in that pocket anymore. So that's why you should start with low molecular weight hit molecules ideally. And same goes for log P and all the other parameters, except for FsP3, because as Lovering noted, there isn't really an upper limit. So let's try to aim for a high sp3 content because it makes your compound already more lead and candidate-like. We need to talk about pains. Pains are pan-assay interference compounds, and they were identified by Bale and Holloway in 2010. And what they did is they compared a lot of literature data on false positives. And they noticed that the structures of all these molecules contained shared moieties, which are the pains. And the pains moieties are, in fact, chemically reactive moieties, sometimes photochemically reactive, and they will engage with many off-targets and therefore heavily interfere with your assay. Nowadays, pains are mostly deleted or taken out of commercial libraries, but I do urge you, once you identify hits, have a look, see if you can identify pains, and if you do, there's one way to put them: the trash can. Going back to this graph, I told you earlier that as we are generating insights in the structural activity relationship, that we need to generate a lot of analogs. And I can't stress this enough. Organic synthesis will be a bottleneck. It will take you time, and you will have to wait for these molecules. And that means that you'll have to pay the people who make it, which will cost you money, but you know, it will always also take time for you to get those data points in. So synthetic feasibility is key. What do we talk about then when we talk about synthetic feasibility? Ideally, we're looking for short synthesis routes with widely commercially available starting materials, ideally cheap starting materials, chemistry that's scalable and which allows you to follow up quickly. A good example of such a route is depicted here below. This is a route which was developed by Jorik, Randy, Eddie, and Rob, my colleagues here, and they synthesized these bifunctionalized imidazole analogs. And what makes this route so attractive is the fact that they generate this advanced intermediate. This intermediate has an ethyl ester, a terbutyl ester. These are called protecting groups. And you can selectively deprotect these leaving groups. So you can install the R1 group or the R2 group in any order which you like. And having such an advanced intermediate at hand allows you to just make this molecule on a multi-gram scale, put it in the freezer, put it in the fridge, and whenever you want to follow up, you can quickly take it out of the fridge, go to the lab, make those molecules in 4 steps. So this really allows you, this route allows you to generate up to hundreds of analogs in less than 2 weeks. The other side of the organic chemistry spectrum is then natural product synthesis, and the chemists in the audience probably agree with me that this is often considered to be the prestige project of organic chemistry. Why? Because natural products are notoriously known to be super complex, hard to synthesize. They'll often take multi-step syntheses, and a route like this, to which our team leader in process chemistry, Ginger Lutteke, co-authored, this took more than 60 steps to generate this molecule, which is a subunit of the actual natural molecule. You can imagine that a PhD may have spent half of their PhD just trying to get here. So that takes easily over 4 weeks. But also something to keep in mind is that with every step that you're adding to your synthetic route, that you're likely to lose some yield, and that will contribute to the overall yield. You see, for this route, the total yield was 9%, which is impressive already for something that takes so many steps. But it also means that you'll have to start with very large amounts of starting material only to get milligrams of product. Can you then imagine that a client comes to us and they say, look, Sam, we've identified this molecule and it's really pretty, has all the desired properties, super potent. Would you mind making 120 analogs on the methyl position? I'm not saying that we can't. And if you're willing to pay us for it, we're very up for the challenge. But you need to keep in mind that if you have a very long synthetic route and you want to make changes to a moiety that's already installed in the first step of your route, that means that if you want to generate 120 analogs, that we have to go through this entire route 120 times. And that will cost you time and money. So if you have the option, please go for the simpler synthetic route that's more available. What would an ideal scaffold then look like? Well, you could argue that it looks something like this. It's a medium-sized ring. It's flexible at this point. We can change every R group at any time selectively. We can selectively link every R group to lock this ring in an active conformation. And every R group selectively influences one key parameter which you'd like to optimize. And of course, you retain your potency, and you can even optimize that potency if you like. Well, such a molecule would really allow you to, make really this, this array, all different combinations to make have this combinatorial enumeration of all possibilities. That's never the case, fortunately. Oh yeah, and all reagents are cheap. That's never the case, but it doesn't mean that we can't aim to make these compounds and can't try to get as close to these properties as possible. To conclude here, I want you to focus on the following. When you're generating your hits, I want you to have a think about what you want. This can be the administration routes, the properties that you desire. You want to have at least a small definition of your desired chemical space. Then you choose the right library for the right target. And then once you have that collection in hand, once you have the virtual file in hand, I want you to look critically at your set and assess whether that set interrogates your desired chemical property space in a good way. Once you've then performed your assay and you've identified your hits, I want you to select the right starting point. These are no singletons, so you have active analogs available, you have initial SAR. I want you to favor low molecular weight, low lipophilicity, and high FSP3. And I urge you to contact the chemical provider, contact the people who gave you that library, and ask them how quickly they can follow up with on-demand chemistry if desired. And the more information you can get about it, the better. If you can get information on the whole synthetic route, that would be amazing. Synthetic feasibility is key. Now, of course, it would be a massive shame to have all this expertise in-house and then not apply it to a compound library of our own. So that's exactly what we did. Symeres has the in-house Symegold library, which is your exclusive, diverse compound library of small molecules. Symegold currently counts over 87,000 small molecule compounds, which are physically made. And the way that we built Symegold was we literally asked all our colleagues, parallel chemists, process chemists, medicinal chemists, we asked them, if you could think of your dream molecule, what would it look like? So we got a lot of ideas, and then we assessed those ideas with our selection committee of experts, and we assessed it according to the 6 Symegold principles. And the first principle is the novelty. Over 60% of our library has no hits on SciFinder. Over 70% of our library has less than 5 hits on SciFinder. So these molecules have a very high novelty factor. The chemistry is tractable. Of course, we didn't go into the lab to synthesize them before we looked at, the proposed synthetic pathways. So That was an initial selection, but synthetic tractability also means that we've looked at short pathways to enable late-stage modification, quick follow-up, as I talked about earlier. Our molecules have high diversity potential, which means that they have at least 2 to 3 exit vectors, so points of diversity, points which you can decorate. Majority of our molecules falls within the Lipinski space, the lead-like, drug-like space. Semigold contains no pains. And we take every opportunity we can get to apply photochemistry, electrochemistry, flow chemistry. We have a subset of macromolecules. We have a subset of natural product derivatives and natural product analogs. So that gives us a very nice coverage of molecular weight and C-log-P. Something to keep in mind is that the line of C-log-P1 is here already, so there's a very large portion of really polar molecules. And we didn't talk about this in depth, but the majority of our set has an MPO score above 4. And the MPO score is it's a measure that gives you an indication of how attractive how attractive your molecule is in terms of blood-brain barrier penetration. And BBB penetration is a lot easier to block than it is to facilitate. So this implies that our set offers a great starting point for CNS discovery, but is definitely not limited to that. I can talk for another half hour about our compounds if you like. I'm going to spare you, but I like to let our molecules themselves do the talking. There's a high amount of spirocycles in here. Medium-sized rings are also present. And if I sparked your interest, reach out. We're very happy to share our catalog, very happy to share a virtual copy of our Semigold database so you can make your own selection, make your own subset, and then you can apply what you've learned today. So very happy to discuss our Semigold set. What I also want to note is, of course, that the additional advantage to Semigold is that once you've identified a hit with Semigold, you're in the prime seat to kickstart your drug discovery campaign. Because Semigold has been made by medicinal chemists, parallel chemists. There's quite a big chance that if you want to have follow-up on-demand synthesis, that it will be performed by that same person who made your hits in the first place. So we have everything in-house to further support you in terms of medicinal chemistry, hit to lead, parallel synthesis, also scale up, also process chemistry, and also early ADME tox studies, and even for oncology targets, your assays. That was everything that I had to say. I want to thank you an awful lot for your attention, and I'm very happy to answer to your questions in the Q&A. And that's where I leave it to Katie.
Sam is being joined in the Q&A section of this webinar by Leon van Berkom and Rutger Volmer, both directors of chemistry at Symeres. And they have a collective 45 years of chemistry expertise that has helped large and small organizations progress their drug discovery programs forward. Leon is responsible for parallel chemistry at Symeres with a focus on leveraging Symeres's library synthesis platform to increase the output of new molecules for drug discovery projects, and Rutger is responsible for analytical chemistry across both our Nijmegen and Groningen sites in the Netherlands. He's also worked at AstraZeneca in Sweden, where he managed structural biology groups with focus on biophysical techniques and fragment-based lead generation. So, on to our first question. How do you know when it's the right time to move on from a hit series, and what does that decision process look like in practice?
So I think in any project, probably the best thing to do is upfront is define the criteria that you want to have for your lead compound. I mean, and people will maybe be aware of the term target product profile, which is used in many companies that do drug discovery, but that often concerns kind of late stage properties like the dose and route of administration, the cost, the shelf life, what sort of patient subgroup you want to treat, etc. But I think if you want to start with drug discovery, it's probably good to have some goals for the sort of short or intermediate stages as well. And this is what we sometimes address with target lead profile. So the profile that you want to have of a compound coming out of hit to lead and going into lead optimization. And they are a little bit more down to earth in terms of the the properties that you want to fulfill. So there will be more around the activity in primary and functional assays, selectivity towards some anti-target, stability in microsomes, physical chemical properties like hydrogen bonds, etc. So really, as I said, a bit more down to earth. And if you are working on your hits, you need to feel that you are improving towards that target lead profile. There needs to be some sort of gradient where you see that that at least with activity and stability, you're not going in the wrong direction, but you are improving those criteria. And you have to feel that you're gonna reach it at some point. But then again, it's when do you give up? That's often hard to say. I sometimes use the term or analogy that say when do you stop searching for a lost purse, right? You never really want to give up. Well, maybe it's there, so you keep on looking. And this also depends on other things. I mean, do you have other equity that you can use? How deep are your pockets financially? Is your company depending a lot on this particular project, or are there other projects that could be run instead? So, people say kill cheap, kill quickly. That's certainly true. But if you have only a few projects to work with, maybe you're not in a position to do so. But I think the main thing is what I said earlier. I mean, you need to see improvement towards a target lead profile. If that's not happening, if you never see any improvement in your potency, or all the compounds that you synthesize are unstable, at some point you have to admit that this is not the right hit to pursue, or not the right project to pursue.
Thank you, Rutger. The next question that's come in, I think, is related to budget and a subset of compounds. So we have a limited budget and can only afford to screen a subset of compounds. What criteria should we prioritize when selecting which libraries to use?
If you're okay with it, I like to take up this one. So, it all depends on the money, right? If you have an infinite amount of money, you want to screen every compound, obviously. But if you have a limited set and you're looking at a diversity library, what I want you to look for is to look for representative compounds. So what we tend to do when we have large sets is we try to identify the core structures. We can cluster sets based on their core structure. And what you actually want to do is, with a limited amount of resources, pick representative molecules for every cluster that is in your compound collection. And when you're picking your clusters, I advise you to always do that with a 2D, a 3D graph of the chemical space that you're covering at that point, so that you're also aware and make sure that you're not looking in the same corner of your box. So what you want to identify there are what are the structural motifs that are shared within one cluster. And once you've done that, I urge you to look at the smallest, most polar compound. But say that you have a little bit more money than that, it can be interesting to pick 3 or even 5 analogs per cluster, because that also gives you, once again, a bit more insight in that SAR data. Plus, you need to keep in mind, if you pick one representative molecule from every cluster, you'll have no insight at the start whether the data that you're getting is just a singleton or whether you do have active analogs. But that's the trade-off. So that's one way that you can start your drug discovery project with limited resources. You start with a representative subset, and once you then identify hits, you will start looking deeper into the active clusters and pick more molecules from that corner of the chemical space.
I can maybe build a little bit on that, although it's not technically what was asked, but I think if you are screening subset or smaller libraries, the quality of the assay that you are using becomes more important, because if you only screen, let's say, 30,000 compound and your hit rate is going to be 0.1%, you're not going to get a lot of hits. So you have to make sure that the hits are really good hits, and that's often an aspect of screening that's overlooked. People, it's easy to focus on libraries because it's very tangible and you can buy them and you see what you have, but the quality of the assay, that's actually being run is also very important and good to keep in mind.
Thank you both. Another question that's come in is, how do you decide on the size and composition of a screening library when you're working with novel or poorly characterized targets?
I think Leon is in the right position for this one.
Can you repeat the question once again, Katie?
Of course. How do you decide on the size and composition of a screening library when you're working with a novel or poorly characterized target?
Yeah, so one thing I've noticed during my career is that sometimes the size of the library is not the key parameter to consider. I think it's more relevant that you have a highly structurally diverse set of compounds, which is way more relevant if you're looking in new chemical space and you want to find a hit compound towards a target that's unknown or with unknown mechanism or an unknown binding pocket with very few relevant hit compounds on similar targets, then I would consider actually the size of the library to be of lesser relevance than the structural diversity.
Thank you, Leon. Is there anything else that Rutger or Sam would add to that point?
No, I don't think so. I mean, I would almost repeat my previous comment, because if it's a poorly or characterized targets are very novel, then the quality of the assay is even more important, because you don't really know what you're looking for. So the accuracy that you get can be quite unfamiliar as well. You know, if you're screening a kinase, then you're looking for certain properties in your hits already, because you know what compounds work against kinases. But if the target is really unknown, then this is already, you can't really use that as a criterion anymore. So you have to be even more sure that you don't have false positive in your hit output.
Thank you, Rutger.
It usually means there's also more work involved in target validation. Because you want to make sure that you have actual binding to your target then.
Okay, we have time for one last question. So regarding the semi-synthetic compounds from your library, where the natural scaffolds are already known from bioactive molecules. So I think the question is asking, do we already know bioactive molecules? Apologies if I've misunderstood that.
So the way that I'm interpreting this question is, when you're looking at natural product derivatives, especially from an IP space, if this molecule, the original natural products, is known to be active, what implications does this have for IP? And the thing is, of course, there's a reason why natural product derivatives are so interesting, because of course, these are molecules that have been optimized by Mother Nature. So it makes them very interesting as a starting point, and to see whether any analogs that we make of them are also active or have even better properties. When it comes to IP space, and Leon, Rutger, feel free to correct me if I'm wrong, but there isn't necessarily a patent on nature. So if you are the first person to come up with very favorable natural product derivatives, and you're the first one to patent it, sounds nice. Now, if this person was talking about that 16-step synthesis towards a natural product derivative, well, yeah, look, once again, we see clients that come towards us with very challenging synthetic routes, and if they are willing to spend a lot of time and money on it and come to us with that question, it's often an indication that it's worth chasing, but it will take time and resources.
Thank you, Sam. So I'm aware that we're at time, so that concludes today's webinar, and I'd like to take an opportunity to thank Sam for his excellent presentation, as well as Leon and Rutger for their insights shared as part of this Q&A session. So thank you all once again for your attendance, and have a great rest of your day. Bye-bye.
Bye-bye.